Nada University, Greater Noida.
KaryoUS, our cross-campus science society, endeavors to foster science literacy amongst its members. On behalf of the team KaryoUS, I extend a warm welcome to another engaging episode of its series of epigenetics. I would now like to invite Ms. Matthew, Head of Connected Learning, SNS, to introduce the event.
Good evening, everyone. Again, for those that might have heard it before, I welcome everyone to what is truly connected learning. You've just been introduced by two of our students from different campuses and one of our students from our Shiv Nadada University. How connected can we be? Anywhere, anytime. It's not just a classroom, and that's what's exciting. We can learn so much more outside of a classroom from experts in their field by collaborating across different boundaries. Again, anywhere, anytime. Tonight's topic is quite enthralling. We owe our biologists, our people that study cells and biology, the creation of the COVID vaccine. But they're looking at solutions to solve problems in the world today for not just humans, but all living things and our environment. So I'm sure you'll find tonight's engagement exciting, but please do take advantage of these learnings. Please look at it as much more than what you'll ever get in a textbook. Having these people at your doorstep is something that not everyone has the opportunity to do. So to Miss Vanita and her great team with our Society of Scientists, I welcome you all and I hope you enjoy tonight's session. Take care, stay safe, let's enjoy what's to follow. Thank you.
Thank you, Mr. Matthew, for your kind words of encouragement. Now, I would like to introduce my school, Vidyagan Leadership Academy. We here create more leaders, and Vidyagan School give opportunity to every child who is curious to learn and have caliber to achieve something in life. I would like to tell also, being a non-bio student, I really found this topic very fascinating and interesting. So I would like to convey my message to everyone that we can learn bio without taking it as main subject. Thank you.
I'm excited to announce that Ms. Ruchi Patra, Ms. Chetna, and Ms. Mala will be mentoring the sessions in the following weeks.
Right. So as I said before, I'm Janvi, and I'm representing our club, Genesis, the Life Sciences Society at SNU. And today with me, I also have our teacher mentors, which is Ishani Roy, a second-year biotech student. Along with me, we also have Bhuvan and Shruti, who are third-year biotech students. And we also have Kartavya and Sandhya, who are fourth-year biotech students. These are going to be the teacher mentors who will be conducting the courses over the next month. And we'll be covering the course Epigenetics. All of this is being conducted under the guidance of Dr. Richa Priyadarshini and Dr. Nandita Chakrabarti, our professors here at Shiv Nadada University. Furthermore, we would like to extend our thanks towards our Vice Chancellor, Professor Ghosh, and our Executive Chancellor, Colonel Gopal, who have helped us gain this opportunity. And also, we'd like to thank the Office of Marketing and Communications led by Mr. Sunil Shetty and our SNU coordinator for KaryoUS, Ms. Vinny Mathur, who has helped us connect all these schools. Through the means of this collaborative course, we hope to ignite the love for science, especially biology, among students and show them how education lies beyond textbook. We're also excited to announce here at SNU, we will be starting a program called Young Mentor Program, which will be following courses like this and two successful modules such as Particle Physics Demystified and Robotics, where all high school children would get a chance to engage in courses similar like this and as this certified science course that we have. And we hope that you guys get a chance to participate in all of these. We would also like to call upon Dr. Richa Priyadarshini to say a few words. Ma'am, will it be possible?
I think there is some technical glitch, so
we're not able to hear maybe. So carry on with next. Yeah.
Arushi.
Yep. Today's session will be conducted by Bhuvan and Ishani. They will be covering the topics mentioned below: the basics of genetics, the basics of DNA, inheritance and Mendelian genetics, and epigenetics. Through the journey of this course, we hope that you get a chance to marvel at nature's complex mechanisms simplified just for you.
These sessions will be followed up with 4 more sessions of certified course, and we really hope that this ignites the love for science that you guys have hiding in there. And we hope that you register for our course. The details and all communication regarding this will be shared with you via email once you register. So please do, and we hope you enjoy the course. And furthermore, we would also like to encourage our participants to put in questions in the chat box, and we would love to answer them during our checkpoints and as and when the course runs. Over to Bhuvan and Ishani to start the course.
um, uh, excuse me. Bhuvan, before you start, may I request somebody to put the registration link in chat box, and probably two of you or three of you be on YouTube, so that if somebody is asking question, you can add this. Over to you, Bhuvan. Thank you.
Thank you, ma'am. I'll just be sharing my screen. Please let me know if it's visible. Okay, so hello everyone and welcome. Today we'll be talking about the DNA, as you know, Arushi mentioned, and apart from that, why we look our parents, right? As the title of the course says, you look like your parents, but why? So before that, let's just cover a little bit about why we look the way we do and why we are the way we are. So let's first begin with what is DNA. DNA is short for deoxyribonucleic acid, and you must have mentioned this word DNA a lot of times in your daily life, right? Like saying you look like your parents, you have your mother's DNA, right? So DNA is basically a molecule, and a molecule is just a group of atoms clustered together, which encodes all the information that makes us who we are. So if you're tall, it's because of the information stored in your DNA. One would say that your DNA is like also an instruction manual or a recipe for the way you look. So everything about the way you are has been written in your DNA for an like an instruction. So on a more molecular level, as we can see like in these slides, we see that DNA is a twisted ladder kind of a structure, right? So imagine having a ladder and holding it by two ends, and imagine twisting it once and twisting it twice, and twisting it as many times as you want, and you'll have something that looks like a DNA. So this is the activity that I just mentioned. Apart from that, the scientific word for what DNA looks like is a double helix, or one would say that it's just a twisted ladder structure. Here I have a simulation which will make you further be able to visualize DNA. Just give it a second, it's loading. Right. So if you can see, this is one strand, right? DNA is a structure that contains two strands wound around each other, right? The two sides of the ladder that run parallel. So this is one strand, and this is another strand, and they are wound around each other in such a manner. Now, another thing about our DNA is that they contain information, right? I just mentioned that DNA contains all the information that makes us who we are. So the information that our DNA contains is stored in the steps of the ladder, that is these places. Okay, so once we go back to the slides, the steps of this ladder contain all the information. So if we unwind the DNA, we will see that it contains some letters, right? What are these letters? These letters are chemical structures which we don't really need to get into at the moment, but let's just say these contain letters, and these letters are arranged in an order to encode information. The four letters that are arranged here are A, T, C, and G. You'll see there's only A, T, C, and G. For example, imagine a computer, right? And you have a computer in which you do everything that you do these days. Now, you know that everything about that computer is written in zeros and ones. In the same way, everything about what we are and who we are is encoded using different combinations of A's, C's, T's, and G's. This sequence of letters is called genetic code. Now let's take a look at where the DNA is in our body. So so we contain cells. We know that humans are made up of millions and millions of cells, right? Cells contain a nucleus in which the DNA is stored. All the cells in our body contain the same amount of DNA, and each cell in our body contains the whole DNA that we need to function, which means that every cell in our body contains all the DNA that we need in our, to be what we are. So cells, the two main components of the cell that we need for this session are nucleus, in which the DNA is contained, and a cell membrane. Cell membrane surrounds our cells to contain within it all the other material that our cell has. Think of it as a water balloon, right? So the cell membrane is the balloon, and everything that is inside it is all the water in the balloon. So within the cell, if we can see, this is a cell split open. This blue thing is the cell membrane. The pink structure is a nucleus, and everything else is a jelly-like substance along with other proteins and other things that are present in the cell. If we enlarge the nucleus, we can see that DNA is present as crumpled up threads, and this structure is called chromatin. So in the cell, all this DNA is present in the form of chromatin in the chamber of nucleus. This is the structure, this is the form of DNA in which is present mostly in our cells. So the following video contains an experiment in which we can take out our own DNA from the skin cells of our cheeks, from our cheek cells. So, okay, let's have a look at this video. Sorry about that. So in the experiment, we used a lot of apparatus and test tubes and funnels and droppers. But in real life, you don't really need to do that. You can use glasses from your kitchen and excuse me, and spoons and things like that to do the same experiment that we saw in the video. Now in the video, one thing that we used to isolate our DNA was detergent. The reason why we use detergent was so that it helps us break open the cell wall, and once the cell wall is broken, all the cell components spill out, including DNA and other proteins and organelles that are contained in the cell. Using alcohol and pineapple juice can help everything other than the DNA to dissolve. So that basically helps our DNA to stand out. When you do this experiment at your home, you can very easily skip using pineapple juice, and in place of alcohol, for alcohol, you can use hand sanitizers or rubbing alcohol because they contain the same thing that's present in alcohols in science labs. Then after that, salt again helped our DNA to precipitate out or stand out so that we could see the DNA. So if you can see in this picture, DNA is contained, DNA contains very thin strands. So it's basically a lot of strands, wound around each other. So let's take a look that how all these strands are packaged inside cells that are so small that we can't look at them with the naked eye. So one cell in our body, and let me remind you, our body contains millions of cells. One cell in our body contains about 6 feet of DNA. So each cell in our body contains 6 feet of DNA. So if we take all the DNA in our body and wind it from the earth to the sun and back to earth, we can do this 600 times until all our DNA is consumed. So if each of us contains that much DNA, you must be wondering how that DNA fits inside a tiny little cell which we can't even look at with the naked eye. So imagine our DNA, the structure of which we have talked about. Imagine this double helical structure or a twisted ladder structure is one thread. Now imagine wrapping this thread around a ball and then the rest of this thread around another ball and so on, right? So this thread around this ball is our DNA and it's wrapped around lots and lots of balls. One thread and ball group is called a nucleosome. But that's not important. What's important is that this series of threads wrapped around balls loops around itself, right? It loops around itself to form a structure called chromatin. And this is the structure which is mostly found in our cells. Now, this chromatin also loops around itself, and then it again these loops again loop around themselves, and after a series of loops later, we find chromosomes. And chromosomes are the most condensed structures of DNA found in our cell. So this makes sense how 6 feet of DNA can fit inside a nucleus. Right? It does. I hope it does. This short 30-second video again takes us from a chromosome to the DNA, and we can try and notice how many times the DNA has looped around itself. Right, you saw that? That was cool. Okay. So we have learned about DNA, what it looks like, and that it contains information for what we look like. But how exactly does that happen? DNA, I'll explain to you again that DNA makes us who we are. It will dictate if we have brown eyes, blue eyes, curly hair, blonde hair, if we are tall, and if our dog is, black or brown. I mean that the dog's DNA will dictate, not ours. So how exactly does DNA dictate features? Through genes. A gene is a segment of DNA. It contains a sequence of A, T, G's, and C's, the letters. And since we have, a really long strand of DNA, tiny segments in that DNA are genes. And each gene contains the sequence that will lead to the formation of a particular protein. Now, where does this protein come from, right? I just out of nowhere use the word protein and you'd be like chicken or eggs or milk. But proteins, there's much more about proteins that we don't know yet. One, everything in our body is almost proteins. If we are tall, it's because of the growth hormones, which are proteins. In today's times of COVID, our antibodies that are formed are because of proteins. Our complexion because of melanin, which is another protein. Our hairs and nails are all proteins. So think of each gene as a recipe to make one protein. If anything in the recipe changes, which means if the if the genetic code of that gene changes, the protein that will be formed will be different. So a change in recipe will cause a change in the protein, leading to change in the function of that protein, leading to change in what that protein does. Proteins are made based on the information encoded in the genes. Again, the sequence of letters dictates how our protein will look. Now, in the upcoming slides, we'll finally talk about why we look like our parents. But first, let's take a few questions. So, okay. Are there questions?
Yes, there is a question which obviously Kartavya has very proactively answered already, but yet Arju is asking, it may be a common question, so I repeat here. DNA is present in nucleus of the cell, but RBCs lack nucleus. Do RBCs contain DNA?
So, mature RBCs don't contain DNA, as Kartavya has very aptly answered. But as our RBC matures, their DNA, they lose DNA, but obviously, hence they don't contain DNA. But apart from that, there are other cells in the environment, which do not contain nucleus. They are called prokaryotes, and they do contain DNA, but it's not localized in the nucleus, and it's present throughout the cell. So that would be it. A mature RBC, so RBCs grow, like have different stages of growth. Sorry, Vinita ma'am asks what a mature RBC is. RBCs obviously have stages of growth, like every cell has stages of growth. And once an RBC is matured and, runs around in our blood, it does not contain DNA. Okay, so over to Ishani.
Hi. So now we come to the topic of we look like our parents, but why? Growing up, we have always noticed that we have some features of our parents, like I may look like my mom or my sister may look like my dad, but we have never questioned this. Why do we look like this? Everyone around us has said that you look like her, you look like him. And sometimes we may not even look like our parents at all. We could look like our aunts or uncles or even our grandparents. So, how do their features, who have no direct relation with us, are present in us, in our body? What makes us look like them? So this happens because these features, also known as traits, are passed on from one generation to another through a process which is called inheritance. We all have heard inheritance when we are reading about property inheritance, right? So in the same way, this information which tells us how we look like is passed on from our parental generation to us through this process. Let's talk about a little more about this. Okay, so genotype and phenotype. So these features that we see, like my hair color or my eye color, these are the physical features, and therefore these are known as the phenotype. But what makes the phenotype look like this? There has to be some information behind it, right? That information is known as the genotype. So the genotype, like the name suggests, is related to gene. Genotype is actually the collection of all the genes in one body, but it could also refer to this information behind this trait, like what makes my eyes blue. So how does this genotype, this particular code, do this? It does through a combination of factors which are known as alleles. Alleles are nothing but variants of a gene. They are different forms, just like the flavors of an ice cream. So here, the ice cream, which is the gene, is the commodity which does not change, but the flavors do, like strawberry or vanilla. So just like that, the gene for eye color will always code for the color of our eyes and not the color of our hair or my eyelashes or my eyebrows. But the combination of alleles, the way different alleles, the way these different forms are present together, they will determine what color of eye I will have, right? And when these traits are passed on, we inherit one allele from each parent. And let's talk about the position of these alleles. So these are present on a particular position on one gene. So like in this picture, we can see in the gene A, so on the first position, we have the gene A and the different forms of this are present on the two different chromosomes. Just like that, each gene has a specified position and these alleles are present on those positions on the different chromosomes. All right. So alleles are also paired together. They never exist alone, but they exist in doubles. So if these alleles are present together in a similar way, if two similar alleles are present, they are known as homozygous. Homo meaning similar, and if two dissimilar alleles are present, they are known as heterozygous. So in subsequent slides, when we are talking about heterozygous or homozygous, it just means they have different alleles or same alleles, right? These alleles, previous slide, Bhuvan. These alleles, next one. Okay, these alleles also have the property of being dominant or recessive. So what does this mean? Just like when we talk, when someone is dominant over the other, they overpower them, right? They overshadow them. So just like that, the recessive allele, the recessive trait is always hidden when it is paired together with a dominant allele. So it's written that in a heterozygous individual, meaning two different alleles, if the dominant allele will always mask the effect of the recessive allele. The recessive allele will only be expressed if it is present homozygously. Just like in this picture, the allele for purple color is recessive and the allele for pink color is dominant. So when these two fertilize and have an offspring, so the dominant allele is present along with the recessive allele and therefore we see only the pink color and not purple. All right. Okay, so now let's come to this. Why do we look like a mix of our parents? So we never look exactly like our mother or exactly like our father, right? We always have mixed features. Why does this happen? I'll just tell you. So in an individual, we have two types of cells. One is a somatic cell, and one are germ cells, which form the gametes. So each cell has two copies of a chromosome. So chromosome, as we learned before, is the tightest packed form of DNA which contains the genes, right? So since we have two sets of chromosomes, naturally we have two sets of each gene. So when gametes are formed, only one set of chromosome is passed on to the sex cells. If they are copied doubly, then we would have abnormal amount of chromosomes, which nature does not allow. So that's why we receive one copy each. Now, um, when we mix two things together, obviously they have a fusion, right? So just like that, these chromosomes also fuse together, and some part of a chromosome is fused with another part, and therefore we see these variations in us, right? So when two X chromosomes are paired together, we have a female offspring, and when one X is paired with one Y, we have a male offspring. And these chromosomes are not the exact copies. They have been changed because they have combined, they have recombined together during the cell division process. Okay. Next slide. Okay. So here we have a father and a mother. So now, how many combinations do you think are possible? How do you think these chromosomes would be paired? So as I said before, one cell gets only one chromosome. So here one cell has X and one cell has Y. So in a similar way, the mother will also pass down one X and one X to each of the cells. So just like mathematics, we can compute all the possibilities of what the child could be, right? So we could have an XX, which would result in a girl, or we could have an XY, which would result in a boy. So this is how these chromosomes are passed on from the parent to the child, which results in who we are. But now let's talk about how traits are passed on. Okay. So growing up, we must have, competed with our friends on seeing whose thumb can bend backwards. We must have had competitions on reaching the thumb back to our palm, or and these traits are very easily observable. We always see that we could have detached earlobes or an attached earlobe, and we also have fun rolling our tongues and making fun of others who don't have a tongue rolling. And freckles and dimples we all find cute, but we never ask how do I get these freckles and dimples? Did I get it from my mom? Did I get it from my dad? So these are some traits which are inherited in a Mendelian pattern. So what does Mendelian pattern mean? It basically means it follows the Mendel's laws of inheritance. So these laws were given by Gregor Johann Mendel. He was interestingly not a biologist or a geneticist, but a 19th century priest who conducted a few experiments to see what would happen. So, what he did was he took a true breeding violet pea flower and a true breeding white pea flower. What true breeding here means is that they have homozygous alleles. They are not heterozygous. So as we can see here, the purple has two capital P alleles and the white flower has two small p alleles. So he fertilized one white pea flower with a purple pea flower to see the results. And interestingly, what he found out was that none of the obtained flowers was white in color, in spite of one flower being completely white. So how did this happen? Why did this happen? So as I said before, the concept of dominant and recessive has stemmed from this experiment. So what the law of dominance says that in a heterozygous individual, the dominant allele will always mask the effect of the recessive allele. So here, the dominant allele is for violet color and the recessive allele is for white color. So when these two are paired together, the dominant allele, the effect of the dominant allele is shown and the effect of the white allele is masked. Only in case the two flowers were white, both recessive, then we would have gotten white flowers. So after the first cross, he decided to self-fertilize the two flowers obtained in the previous cross, just to see what would happen in the next generation. So now what he found out was, along with purple flowers, he also got a white flower. But there was no white in the previous generation, right? So why did one white flower appear in this cross? So this happens because during cell division, these alleles are not attached together. They can separate. So these alleles separate into one of the gamete cells, and these gametes can combine in all possible combinations. So here in the capital D, one D is passed to one gamete and the small d is passed on to other gamete, and these two gametes, when they fuse together to form the offspring, the zygote, we get a genotype, which is a heterozygous genotype. So here the cross explains it very clearly. So when the dominant ones are paired together, or even in a heterozygous, when the dominant allele is present, we see the violet color. But there is one possibility of the recessive allele being paired together with the recessive allele. So this is why we get one white flower out of those four. So in this cross, we can see the ratio is 3:1. It doesn't mean that Mendel also got 3 purple flowers and 1 white flower. He could have gotten like 75 purple flowers or 25 white flowers, but the ratio is maintained. In cases when the ratio is not maintained, it just means that something has gone wrong with the flower or during the process of fertilization. But this is the foundation for uh, these possibilities of phenotypes and genotypes. Okay, next slide. Okay, so after this, Mendel was not satisfied with just 2 crosses, so he tried to do a third cross, and now he selected 2 traits. One was the pea shape and one was pea color. So what he did was he took 2 seeds, one was round and yellow and one was wrinkled and green, and he decided to cross them. And what he found was that all seeds were round and yellow, and not one seed was green or wrinkled or did not have any recessive characteristic. So just like before, he again self-fertilized this first parental generation and he found out another cross. Okay. So now in this cross, he got varied results. He got wrinkled and green, he got round and wrinkled, he got various um, phenotypes and genotypes. But how did this happen? So one very distinct difference between the previous cross and this cross is that here we have 2 genes. In the previous one, the gene was only for the color of flower. But here we have gene for pea shape and pea color. So why weren't they linked? So when these were crossed, shouldn't one gene influence the other gene? But actually this doesn't happen. So think of it like this, like how when twins are born and when twins grow up, everyone expects them to do everything together. Like they would wear matching outfits, they would go everywhere together, they would do everything together. But this doesn't happen, right? They do so independently, irrespective of the other person. So just like that, here too, one gene doesn't affect the other gene. So how the capital R allele is inherited doesn't influence how the Y allele would be inherited. They could pair with any allele in any way. So that's why we have so many combinations, and this gives the law of independent assortment. Basically, what it says is that these alleles would separate independently, irrespective of each other. Okay. So right now we have covered Mendelian pattern of inheritance, and single gene inheritance just means that one trait would be controlled by one gene. Like we saw, P color was controlled by one gene, P shape by one, and so on. But there is also another pattern of inheritance, which is non-Mendelian. Basically, it doesn't follow Mendel's laws, it deviates from it. And some types of this pattern of inheritance are the following: polygenic traits, multiple allelism, and codominance and incomplete dominance. So let's talk about these in the further slides. Okay. So incomplete dominance. So what does incomplete dominance mean? Just like the name suggests, it is a midway compromise between complete dominance and no dominance at all. So here, none of these alleles are dominant over each other. They compete together, but they don't show their effect completely. So here, when a white snapdragon flower was crossed with a red snapdragon flower, one would expect that we would get all red, right? Because of the Mendelian laws that we just read. But this doesn't happen. What we actually obtained was pink flowers, which was a mix between the two. So that's why it is an incomplete dominance. It is not completely white or completely red, but it is midway between the two. There's also a concept known as codominance. So as the name suggests, in codominance, the alleles exist together and they are happy to exist together. So they don't, so there's no compromise between them. They exist independently but together. So in flowers, as we can clearly see, this is a cross between a red and a white flower. But this is not a pink one. This has clear, distinct colors of red and white in the petals. This is because the R for red and W for white exist together codominantly, just like in humans in blood group. So you may have noticed, like some people would have their parents would have A group or B blood group, and you may have O blood group. So why did this happen? Like my mom didn't have A. So how my mom didn't have O, my dad didn't have O. So why did I get O? Or maybe I could have got AB, right? This also happens because the alleles for A and B exist together codominantly. They are not overpowering or overshadowing the effect of one, but we can see both A and B. And in the previous slides, we saw only two alleles for one gene, right? Like the purple color had capital P and small p, like capital W, small w. But here we are seeing six types of alleles. So there's I, capital A, IA, and or small I, IB. So there are so many alleles, which is also a deviation from what Mendel said. Mendel, Mendel said that there would be two alleles for one gene, but here there are six alleles and they are existing codominantly. Okay, next slide. There is also another deviation, which is known as polygenic traits. So poly meaning many. So here one trait is controlled by more than one gene, unlike to what Mendel said. So as we can see here, skin color is controlled by three genes which have two alleles each. And when these gametes are crossed together to see the possibilities of the genotype and phenotype, we have 64 possibilities. Just imagine the variations in skin color we have. So now that you know this, that's why we have so many different shades of skin. Someone may be a light brown, someone may be a little wheatish. Just like that, human height is controlled by more than 50 gene and it has 700 gene variants which control human height. So we may look the same height, but it actually differs by a millimeter or so, which is not easily visible to us. So unlike the single gene inheritance, here one trait is controlled by a number of genes which influence even the smallest fraction in the trait. So this covers non-Mendelian inheritance. Um, so do we have any questions regarding these topics?
Many questions. I think someone should read those questions. Otherwise, I'll read. It's in the.
Sure, ma'am. One of the questions that is being asked is, can three and four alleles act together?
Yeah, so just like we are seeing here in this the cross for skin color, we have three alleles, three genes, and there are six alleles. So what we are seeing is three alleles acting together, and here it is a quantitative assessment, right? So look at this. We have all capitals, so that's why it is the darkest shade. And look at this one, the last square, which is the white shade. Only the small recessive alleles are present. So these are the dominant and recessive one. But look at the middle alleles, right? So so the skin color here is determined by the number of capital alleles or the number of small alleles. Those possibilities which have more of the smaller or the recessive alleles, they have a lighter shade. But those which have more number of alleles, like more capitals, like capital A, capital B, they have a darker shade. So when three or four alleles act together, it is determined quantitatively. So if there are more dominant, it would inch towards the dominant trait. If there are more recessive, it would incline towards the recessive trait.
Is there
Are
some more
there
question? Otherwise, you can go for.
Yes, ma'am, there is one more question. How are twins produced?
okay? So twins, the so when the cell is fertilized, right? When the gamete from the mother and the gamete from the father are fertilized, when they reach the womb, sometimes they divide, and that's what gives rise to identical twins. So, because the same cell is dividing, that's why we have two twins. But there can be another type of twins also when two egg cells are fertilized, right? So that's also when we have two cells. So the twins can be formed in different ways. So these were the basic ones. Okay, so
Okay, so I think the rest of the questions have been answered on the chat. So we'll be starting with a brief introduction to epigenetics. Now, we all know what these cells are, right? The first picture shows heart cells, the second picture shows red blood cells, and the third picture shows brain cells. Now, all of these cells have at one point in their life contained the same amount, the same sequence of DNA that we contain in our body, right? Then, if all our cells share the same DNA sequences, then why do we have different types of cell types? The answer is that there's one more factor that impacts gene expression, which is epigenetics. So modification of DNA without a change in the genetic sequence is called epigenetics. Let's learn a little more about it. So the third type of genetics after Mendelian and non-Mendelian genetics is called epigenetics, where epi means above and beyond. And the reason why we say epigenetics is beyond genetics is because the changes which are a result of epigenetics take place without a change in the DNA sequence. So changes in the phenotype that take place without a change in the DNA sequence is called epigenetics. The main two reasons why epigenetics is needed in the body is one, to package DNA, as we learned in the previous slides. DNA packaging and the process of condensing DNA into a small place is facilitated by epigenetics. The other reason is regulation in gene expression. As we saw how all those cells were very different from each other, the reason was some genes, because of epigenetics, had been turned on and some genes had been turned off. So only a number of important genes were being activated and expressed in order to uh, make the cells the way they are. So this picture shows a nucleus which contains DNA, and this DNA is of two types here. The dark blue kind, heterochromatin, and the light blue kind, euchromatin. And the difference between these two types of DNA is how tightly they are wound. Imagine taking a thread again and imagine crumpling it up to make it a very, tight mass. That would be heterochromatin here. And if we, intend to open up a thread a little and loosen it out, that would be euchromatin. On a more molecular level, if you remember DNA being wrapped around balls, this would be heterochromatin, the tighter one, and this would be euchromatin. So, minor differences between heterochromatin and euchromatin. Euchromatin contains the word EU, which means true. The reason why euchromatin is called true chromatin, because this particular DNA strand contains genes which are active, which will code for proteins and which will lead to results. Whereas heterochromatin, since it is tightly bound, it won't be producing any protein, and the genes present in this segment of the DNA are turned off. So, okay, in the next slide, we talk a little more about heterochromatin and euchromatin. Okay, so we can see very clearly here that the DNA in this picture is tightly wound around each other, so we would call it heterochromatin. And here the DNA is loosely arranged, and we would call this euchromatin, right? Euchromatin, as this color indicates, contains active genes, and heterochromatin contains genes which are turned off. Now, since we all know DNA is a bunch of chemicals, right, stuck together performing a functions, in the same way, a methyl group, which is another chemical, gets attached to the DNA, which makes the DNA, crumple around itself and condense around itself to form heterochromatin. Whereas if we attach another chemical called acetyl group to the DNA, it opens up and starts, containing genes that are active. This here this text says DNA is accessible to other proteins in the cell and hence the gene is active, whereas DNA is inaccessible in this region of the chromatin and hence the gene is inactive. So the two changes which lead to the formation of heterochromatin and euchromatin respectively are methylation and acetylation. We have understood the use of methylation and acetylation, and we will be talking about it in more detail, and you don't have to remember any of it for now. In the further sessions, we will be elucidating upon all these concepts and a lot more. So, bees, right? Just like all the cells in our body, bees are also clones of each other. Clones means that all the bees in a hive have the exact same genetic sequence. And there are 3 types of bees in a hive. First one, queen. Her job is to lay eggs, right? This is what a queen does. A worker bee guards the hive, takes care of the children, baby bees or larvae, and collects food and water for the hive. And a drone bee, which is a third kind of bee, contains only males, and unlike the queen and the workers, it does not have any stinger. So, sorry about that. Workers are females, drones are males. So the reason behind, having the same genetic sequence and still being so different from each other is again epigenetics. In queens, all the genes are active. Every gene in the DNA is active, whereas in workers and drones, some or the other genes have been turned off so that the workers and the drones can perform their functions and help out in the hive, however way they are needed to. So a few more examples. One, this kind of a cat is called a calico cat. And take a look at the coloration in its coat. Now, all calico cats are only females. The reason behind this is also another phenomenon that is related to epigenetics. This will be covered more in detail in the upcoming sessions. And these are identical twins, as you can see, and their hair color is different naturally. And not only just their hair color, the way they behave is also different, as we know from our life and around us. We know that twins are very different from each other. Identical twins contain the same genetic sequence. As I have repeated about 5 times now, the answer lies in epigenetics, and we will talk about it more in the upcoming sessions. Thank you. So any more questions? I'll be happy to answer.
Wow, this was really a very wonderful session. If you have any more questions, please put them in the comment box. And thanks Ishani and Bhuvan for coming up and finding a way to make it communicable and easy to understand.
Thank you.
As you can see that the DNA is not what it seems, and there's so much more to understand on a genotypic level, as you just learned. Just to remind you that the registration link for the certification course that will run over the next month is in the chat box, and all details for these courses will be sent to you via email. So do register, and we're looking forward to meet you. The courses will be interactive, where you can interact with the mentors one-on-one and ask them questions as and when they're teaching. So please do register. And again, to remind you, there will be sessions like this, and keep an eye out and follow all pages for Shivnada University, Shivnada School, the Engage page, and even our Genesis page to find out more updates about upcoming courses. And keep an eye out in your inbox in the month of October for more fun courses that we are planning for you.
Now I would like to call Malamam for delivering the word of thanks.
Am I audible?
Yes.
Okay, first of all, this was an amazing session on genetics. Everybody who's here listening to the wonderful presentation that our SNU students have brought in. They have really worked very hard on all this to bring this to you. This is just the introduction. Stay tuned for other sessions as well. As Janvi just told you that there are registration forms. Please do register for the upcoming sessions. To unfold some more mysteries about genetics, why do we look like we do? Why don't we look like our parents or our siblings? So these, similarities and differences, and there are a lot of questions that I see about twins. So stay curious and keep bringing in your questions in the further the following sessions that we have. now we are concluding this particular session, and thanks are due to a lot of people. First of all, Mr. Matthew, we thank you a lot. We know that we always have your solid support. Your inspiration is always there for us. So thank you so much. Ms. Vinita Sharad, of course, she is the leading light of this entire program. Then our mentors at the SNU University, Shivnadar University, Professor Richa Priyadarshini and Professor Anandita Roy. You have been mentoring the children very well, and they have taken these sessions very well. Then we have our student mentors from the Genesis, which are Janvi, Ishani, Bhuvan, Kartavya, Sandhya, and have I called everybody's name? I think I have. So particularly Bhuvan and Ishani, you have done a wonderful job of introducing genetics to the children. And then our student compeers from SNS Noida, Arushi Rai, and Priyanshi from Vidya Gyan. Thank you so much for taking this program forward today. So stay tuned for the future sessions. Thank you so much, everyone. Vinita, would you like to say something now?
I just want to thank a big thank you to Naman, who is part of each and every session. Almost every week, 2-3 sessions are going on, and he has to stay there for all technical support.
Yeah.
Thanks to Suhana also for
technical team, Suhana and Naman and Satvik, you have done a wonderful job of keeping this entire program together, doing the, that is the, they work in the backgrounds to make all this happen, and they keep it together wonderfully. So without their technical support, all of this won't be possible. Thank you so much, children.
And a big clap to Mala and Chetna and Ruchirata and all the students who have taken sessions. A big thank you to Sandhya Kasi also, because it all started first with, and then it was. Thank you very much.
Thanks, everybody. Just stay tuned and stay curious, and we'll work together.
And get registered for the course.
And definitely register for the courses so that we can take it forward.
Yeah. Also, Mala, I hope everyone knows that will be a certified course. The certificate will be given with the signature of one of the professor mentor from SNU, and obviously as a STEM coordinator. So this is important that children get registered for further so that we can take it further. Over to you, Naman.